The endocrine cells of the pancreas make up just 2% of its mass, but are organized into over 1 million discrete clusters called the Islets of Langerhans. Paul Langerhans discovered these patches of endocrine tissue in Germany in 1869. Each islet is encapsulated by connective tissue and contains several thousand cells belonging to 5 major cell types, each of which produces a specific type of hormone. About 60% of the cells are insulin-producing beta cells, which form the central core of each islet. Insulin is a peptide hormone that stimulates the liver, adipose tissue, and skeletal muscle tissue to import glucose, thereby lowering the glucose concentration in the blood. Unlike lipid-soluble steroid hormones that connect directly within the nuclei of target cells, amino acid-based hormones interact via receptors on the cell membrane. However, insulin receptors are present in nearly every mammalian cell and are required in order for cells to import glucose across the cell membrane. Insulin is initially created in the precursor form pre-proinsulin and then converted to proinsulin. Proinsulin is cleaved by C-peptide and stored in granules. The presence of high glucose levels in the plasma triggers depolarization of the cell membrane and the release of insulin from beta cells. Alpha cells are the second most common islet cells at around 30%. Alpha cells secrete glucagon, another peptide hormone that has the opposite effect of insulin and increases the concentration of glucose and fatty acids in the blood. The remaining 10% of islet cells consist of delta cells, epsilon cells, and pancreatic polypeptide cells. Delta cells release somatostatin, which acts to limit the release of both insulin and glucagon. The final 2 hormones also act in opposing directions. Ghrelin, secreted by epsilon cells, stimulates appetite and promotes fat storage, while pancreatic polypeptide promotes satiety by inhibiting the release of gastric acid and slowing the movement of food out of the stomach and through the upper intestine. The pancreas tightly regulates and coordinates release of hormones using negative feedback loops. Insulin suppresses release of glucagon and vice versa. Parasympathetic and sympathetic nerve fibers enter the islets through neurovascular bundles, and islet cells communicate with each other through gap junctions. Diabetes mellitus results from improper regulation of blood glucose levels. Diabetes mellitus type 1 is an autoimmune disease in which immune cells target insulin-rich beta cells, increasing the risk of vascular and kidney disease, diabetic retinopathy, and neuropathy. Plunging insulin levels can disrupt the ability of cells to use glucose, triggering the release of free fatty acids from adipose tissue. The liver oxidizes these fatty acids to form ketone bodies as an alternative lipid-based energy source that does not require receptor-mediated transport across the cell membrane. However, ketonemia, or accumulation of ketone bodies in the blood, causes diabetic ketoacidosis and possibly diabetic coma. Diabetes mellitus type 2 is more common and is associated with insulin resistance, in which long-term elevations in blood sugar level lead in turn to long-term elevations in insulin levels. This causes cells to become progressively less responsive to insulin, prompting the pancreas to further increase insulin production, eventually leading to exhaustion and impaired function. The term pancreatic cancer is often synonymous with pancreatic ductal adenocarcinoma, but pancreatic neuroendocrine tumors represent a small but important subset of pancreatic cancers. Unlike exocrine tumors, endocrine tumors are likely to be well differentiated and tend to grow more slowly. The 5-year survival rate is 65%, compared to only 5% for pancreatic adenocarcinoma. Depending on whether the affected cell type continues to secrete its signature hormone, neuroendocrine tumors may be classified as functioning or non-functioning. Most neuroendocrine tumors are non-functioning and are diagnosed incidentally. However, 60 to 90% are malignant. Functioning neuroendocrine tumors differ greatly with respect to which cell type is affected. With enough supplementation, it is possible to live without a pancreas, but the process involved underscores the vast number of essential functions performed by the pancreas. A portal system within a portal system, the Islets of Langerhans monitor and finally balance the energy needs of the body. This unpreprocessing organ, once thought to serve only as a cushion for the stomach, provides a vital link between the food we eat and the energy used within our cells.